All right, Taylor, are you ready to get started?
I am indeed.
All right. Welcome, everyone. Thank you so much for joining us this evening. My name is Cinnamon Moffett. I'm the Research Program Manager at Hatfield Marine Science Center in Newport, Oregon, and I will be your host this evening. Hopefully you have your favorite food and drink all ready for you, and we'll get started here in just a second. A couple of logistics. This is a webinar, so your mics and camera and screen shares are disabled, but you will be able to communicate with us through the chat box at the bottom. You're welcome to type your questions in at any time, and we'll get to them at the end. The other thing I wanted to just do a little promo for our next Science on Tap, which will also be virtual. It will be June 25th. Angie Dewar is going to be talking about fishing fleet here in Newport, Oregon. So we're excited to have her join us. But for the main event and what you're excited about for tonight, let me introduce today's speaker. For over 15 years, Taylor Chapple has been studying sharks and other large marine predators around the world. His work uses cutting-edge biologging tags to quantify the animal's movements and describe the trophic and ecological interactions of these top predators. He has a PhD from the University of California, Davis, and in 2019, he joined the Oregon State University as an assistant professor with the Department of Fisheries and Wildlife. He is based out of Newport, Oregon at Hatfield Marine Science Center, working with the Coastal Oregon Marine Experimental Station. And his work now focuses on the sharks of Oregon and their effect on local coastal ecosystems. So Taylor, I'll let you take it away.
Great, thanks, Simon, and welcome everybody. Would have been really nice to meet all of you in person, but I guess this is where we are now. I hope you all have your, if you're of age, have your pub drink with you, and if you're not, I hope you found something equally as delightful to help you get through this. I just wanted to talk a little bit about some of the work that I've been doing over the past 15 years. I think Cinnamon introduced it. I've been studying sharks and other big fish. And so tonight I'm going to talk about white sharks. And this is some work that I've been doing in California and in South Africa. And I guess just some general housekeeping. If you have questions, feel free to jump on the chat and send questions. If there's things that are specific clarity that you want for different sections or things I talk about, please send a chat over, and Cinnamon will interrupt me if I don't notice it. And I'll also answer some more general questions and things towards the end. So with that, let's get started. So the title of this is a little bit different than what was in the announcement, and it's all along the same lines. And the idea of what I try to do as a scientist is to learn and take that knowledge and share it. And so my goal is to get everyone is excited about the outside world, and in this case, the predators that are outside our back doors, and then take the information that we learn and use that to understand them so we can appreciate them. So with that, normally this is sort of what I think you or most of us think about or would think about when someone would say shark. And it's the idea is that they are these big predators, and their teeth and they're bloody and gory, and there's all these scary sort of feelings that you get when you think about sharks. But my goal is to change the way that I think about sharks and the way that we all think about sharks, and think about more of their role in our healthy ecosystems and how really complex and amazing these animals are. And so a lot of this stemmed from this iconic scene, which many of you are probably familiar with, the iconic film Jaws. And so this was what really got into the psyche of everyone, and it really sort of changed the way that generations looked at sharks and looked at the ocean. I know folks that were nervous to go into pools because of what they saw on Jaws. So again, my goal is to change that, because the truth is that you're more likely to die from a selfie than you are to be attacked by a shark. And so there's very few people that walk around terrified of selfie sticks and cell phones. But the truth is that's much more dangerous to you in your day-to-day life than a shark attack is. But I have not seen Selfie Week as a something on Discovery Channel that you can watch. So far, it's still Shark Week. The truth is that most of us are going to interact with sharks in a much different framework. We're going to interact with them somewhere, either in cosmetics. So that's things down in the bottom left here, which are cosmetic products based on sharks. Cartilage that folks once thought could help fight cancer, and it's more of a placebo. It was folks thought sharks didn't get cancer, so by taking cartilage pills that could help you, and it was a little bit more of a sort of a snake oil than it was actually something that would help. And then shark fisheries, and so you're probably familiar with this picture on the bottom right, which is shark fin soup, and that is a huge driver of shark consumption in the world. About 70 million sharks a year are killed. At least 70 million sharks a year are killed for the fin trade. So in truth, they need to be much more worried about us than we need to be worried about them. But there is a risk of shark attacks. And so this is a figure that shows on the bottom axis, you can see the dates starting at 1960 going through 2012. And on the y-axis on the side is the number of attacks. And what you can see is that over time, the number of attacks have changed. They've increased. And so the blue is the United States and the red is other countries all put together. So you can see there is an increasing number of attacks. But is that the whole story? So what we did is we got together and we said, well, let's figure out what the story is. Is it really that the number of attacks globally are changing and shark numbers are increasing and this is something we should be worried about? So we went down to California, where I do a lot of the work, and we looked at white shark interactions. And we documented back to the 1950s number of shark attacks that occurred. And we saw that was actually increasing, similar to the global trends. And so on this figure in the top right, again, you have time along the x-axis, so 1950 till about 2014, and on the y-axis is the number of attacks. And if you look at the dotted line in the background of that figure, you can see that line is slowly creeping up. So it is increasing. But there's a lot of factors that play into that. And so we wanted to think about what those factors were. And the biggest one of all was people. And so we see during that same period of time from 1950 to 2013, which is this next little figure, the number of millions of people in California was growing at a great rate. And so we wanted to see how these different factors affected the way that we perceive shark attack risk. So there's going to be a few figures on this next slide. I'll talk through them, but it's just to get a general sense of what we did. And so we looked at four different ocean activities. Scuba diving, you can see a little picture on the right side of the activities. So scuba diving, surfing, swimming, and abalone diving. For those that are not familiar with abalone diving, basically you're snorkeling and diving down and picking a small mollusk off of rocks. And it's an activity a lot of people in California do. And so if we look at those four different activities and the number of people that are involved in those activities since the 1950s, those numbers have gone up almost exponentially, but precipitously in that same period of time that we're talking about in increasing shark attacks. So if you look at the figure on the left side, those are those different activities through time. So from 1950 or 60 through time, you see all those activities, whether you're scuba diving, surfing, swimming, abalone diving, all of those are going up. So once we started to take that into account, what we found out was that your probability of being attacked is going down. So this is a figure that shows time, again, over the x-axis, and the number of million surfers in your risk. And so if you just follow the general trend of those colors, that's the direction. So basically, if you started to take into account the number of people in the water, your risk of ever interacting with a shark in a negative way was going down precipitously, going down by almost 90% over the last 50 years. And that was a case that held up for all of these different activities. So whether you were scuba diving, surfing, swimming, abalone diving, once we took into account the people that were in the water, the new number of people, the increasing number of people, your actual risk was going down. So the idea that shark attacks are going up and we should be afraid of going in the water is not necessarily the case. The sheer number is going up, but your probability is actually going down. So that's a little bit of background about sort of our interactions with sharks. And now I'll give a little bit of background about white sharks in general before I get into more of the research that I do. And so globally, we know that there are 6 distinct populations. And so these are genetically distinct. So they don't interact, they don't overlap. Animals from different locations don't meet one another. So starting from the top left of the figure, that's the Northwest Atlantic. So that's the eastern seaboard of the US. And then in blue, you have the Mediterranean. Further down south is South Africa. We have the Australia and New Zealand, and then the Northwest Pacific and the Northeast Pacific. And so I'll be talking mostly about the Northeast Pacific. And the reason I have the 2 in blue are those populations that we know almost nothing about. But the work that I've been doing is mostly in the Northeast Pacific, as well as some work in Cape Cod on the East Coast, and then down in Australia, down in South Africa. So white sharks are live born. They're somewhere between 1.2 and 1.5 meters. So that's about 4 feet long when they're born. And a female will have anywhere between 3 and 14 pups. We think it's probably somewhere in the realm of 8. So baby shark is called a pup. And they are regional endotherms. And this is what makes them incredibly unique. And so most fish are, I guess, like a snake, where they're basically cold-blooded. They are the temperature of their ambient environment. So white sharks are different. They can heat certain parts of their body, and they actually take their red muscle, which is really the working muscle. So you imagine if you're eating a chicken, the legs, the thighs, so those are the red ones. So those are the ones that are working all the time. And then they have the white muscle, which is the breast muscle. But sharks put the red muscle on the inside that's making the heat, and then they surround it with all their white muscle. So the red muscle is right in the inside of their core, surrounded by white muscle, and that works as a buffer. So that allows them to maintain their heat. They also heat their eye, and they heat their brain. And this is really important for them because it allows them to exploit fast-moving marine mammals in really cold water. So if you were a shark that wasn't able to elevate your body temperature, elevate your metabolism, you'd be really sluggish. So your chance of catching a really fast elephant seal or sea lion would be really slim. So it's this endothermy, this regional endothermy, that allows them to really exploit these fast-moving marine mammals and be the really incredible and efficient hunters that they are. They go through what we call an ontogenetic shift, and that happens about 2 to 2.5 meters long. And what this is when they're small, when they're born, they're small, and they are feeding mainly on sharks, other sharks, and fish, cephalopods, things like that. And they're in warmer waters down south. For us on the West Coast, they're down in the Southern California Bight, which is basically the Santa Barbara, LA, San Diego area, the warm water there. And then once they hit about 2.5 meters long, they go through this ontogenetic shift where their dentition changes. They have this thermal inertia. Basically, they've gotten big enough that they can maintain their body temperature even in cold water. So now they can start to venture into California, Central and Northern California waters that are much colder. So somewhere in the realm of 40 and 50 degree water, as opposed to 70 degree water down in Southern California. So this ontogenetic shift is that really critical time where they basically graduate from eating fish and squid up to eating marine mammals. And so the work I'm talking about today will be after that ontogenetic shift, because the techniques that I use to tag them utilizes their predatory strategy on marine mammals. So I'm just talking about the animals after about 2.5 meters long, which we call the subadults and adults. Females gestate for somewhere between 12 and 18 months, and we think they probably take a rest year. So that means that at the most frequent, they would give birth every 2 years, but we think it's probably a much larger span than that. The populations of each of these different genetically distinct populations are low. They're somewhere in the hundreds to low thousands once you take into account all of the animals within that population. So that's not the global estimate of abundance, but that is the estimate of each of those distinct genetic populations, the 6 that you see at the top of the screen. So they're somewhere in the hundreds to low thousands. Their maximum age is somewhere around 70 years. And this is pretty cool. This is something we just came up with recently. And we thought 5-10 years ago, we thought that sharks only made it to about 35 years old. But now we know it's upwards of 70 years old, which is a really cool recent finding. And that makes a lot of difference for how we understand their life history, about how their strategy to survive and how we can continue to protect them, preserve them as a species. And we used bomb carbon dating, which is very similar to what you do to date fossils or to date mummies and things like that. That same technique, very similar technique we use to age these animals. And they do these really incredible large-scale migrations, which we'll talk about in a few minutes. But I do want to point out the question marks that I've put up here on this screen. And that's because there's so many facts about these predators that we still don't really know. We have ideas, we've inferred from different little pieces of data, but a lot of information we don't know for sure. That's because it's incredibly difficult to study these large, cryptic, migratory animals that live in the oceans. So it's a struggle to get these down to real hard evidence, but these are the best estimates that we can come up with. So the work that I do, that I'm going to present today, is some work that I've been doing in California since about 2004, and then work in South Africa that started in 2013. So in California, I work in 3 different sites. This is Tomales Point, which is about 35 miles north of San Francisco, part of the Point Reyes National Seashore. The Farallon Islands, which are about 25 miles west of the Golden Gate, and Año Nuevo, which is an island. It's about 30 miles north of Año Nuevo. Is about 30 miles north of Santa Cruz, and about these 3 locations make up what's typically called the Red Triangle. And so these are the 3 locations that I work in California. I also work in South Africa, and this is a spot called Kansbai, which is basically just west of the southern tip of Africa, and it's a place called Shark Alley. And if any of you are Shark Week aficionados, you'll know Shark Alley and Dyer Island is very famous shark spots in South Africa. And so the way that I normally go about their research is we use their visual hunting strategy. So white sharks are incredibly good at visual hunting. They use their eyes to find their prey, so they'll swim down deep. Their dark color on top blends them in, camouflages them to the bottom. They look for a silhouette of their prey at the surface. So we basically exploit that technique. And so we put a decoy at the surface, which is what you see here, which is basically just indoor-outdoor carpet in the shape of a seal. That's what it looks like when it starts, and that's what it looks like a few weeks later. And so the idea is that we're trying to entice the sharks to express their normal predatory behavior. So the sharks will come up and bite and investigate this decoy. And it's soft, so it doesn't hurt them, and it's pretty resilient. We go through a lot of zip ties, but the decoys last for about a season, so we have to retire them. We also use a little bit of marine mammal baits, and this is really important because the sharks, once they come up to investigate the decoy, they're also very shy. They're very cautious, I should say. And if they come to see the decoy and they notice the research boat, they'll leave right away. They may think it's a bigger predator or a bigger shark. So we use a small piece of marine mammal blubber in the water to create a localized scent. And so this isn't a big chumming episode that you would see on TV. This is a very localized scent that just puts a smell around the boat. And that's to keep the sharks interested so that they can make a few passes around the boat so we can collect data. Those data are usually comprised of photo IDs, which is taking the dorsal fins, and I'll talk about that in a second, and then external tags. Again, I'll talk about those in a few seconds as well. So once the animals are around the boat, they smell that, they come around the boat, and that's when we have the chance to put the tags on and get the IDs. And so this is a picture in California of some of the team from Montana State and the Monterey Bay Aquarium in Stanford that I work with to put this is a biologging tag that I'm trying to put on the dorsal fin of their shark as it's coming towards the boat. So one of the first questions I had when I started studying sharks was to find out how many of these animals were off our coasts. And I sort of gave it away a little bit earlier, saying that the numbers of the global populations. But but I'll get into it a little bit more now of how we actually did that. And so, as I said before, the dorsal fins of white sharks are basically like a fingerprint. And so they have these notches and peaks in the trailing edge of the fin so the dorsal fin is the fin on their back, and the back edge of that is unique, and it's maintained for long periods of time, upwards of 35 or 40 years at least. So we can think of it like a fingerprint. Some people think of it as a barcode. So every time we see an animal, we can take a photograph of that animal and then compare those over years, and we know when an animal shows up and how long that animal's been around and how many new animals there are every year. And so some sort of fancy math, we can come up with a population abundance. And so just to prove to you that these fins do look the same, this is a shark that was photographed 5 different years. And you can see with the arrows that all of those notches are almost exactly the same. So that animal has not changed that trailing edge during that 5-year period that we photographed it. So this is normally the time I ask people, well, how many animals do you think there are? I can't ask you all that, but so I'll just get to the punchline. We'll find out that in Central California, which makes up a large portion of that whole Northeast Pacific population, that there's only about 200, 220 of these subadult and adult white sharks. And for us, that was a really surprising number, I guess mostly because we didn't know what to expect. But we're still working on whether that is a healthy population number or a population that's in danger. Again, this is just a point in time. So we're just saying this is what it was at one point, and now we have to continue to study them through time to see if that point increases or decreases or stays stable. So that's a little bit about the abundance information. Now I'll talk about some more of the movements and energetics and behaviors of these animals. And so typically the way that we understand sharks and some of these cryptic animals in the ocean is that we have an interaction. So like this picture, this is us out attracting sharks at the Farallon Islands. And typically what's happened is that we say, all right, we interact with this animal for 5 or 10 or 15 seconds, and we're going to infer what it does the entire rest of its life and its life cycle and its decisions and how it feeds and all these things. But that doesn't really work. It would be like following you to the coffee shop in the morning and seeing you stand in line for 2 minutes and then going home and writing a whole report about what you do for your entire life and everything there is to know about you, which doesn't work because that's a sliver of time that we're trying to interpolate the entire lifespan and the entire yearly behavior of these animals. And so I just sort of break this down as taking that moment down to that small point in time of your day. So what we needed to do is be able to travel along with these animals, to see their environment with them, to be on board with them as they make their yearly migrations or their movements or their decisions on whether to eat a prey or not to eat a prey. And so one of the technologies that came about is called the biologging tag. And this is an incredibly cool tag because of the amount of data that you can get from it. What it houses is a camera, so we can see what the animal sees. We can see how the shark interacts within its environment, the type of prey that it sees, the type of habitat that it's in, the water clarity, things like that we weren't able to see when we just interacted with them opportunistically at the surface. It also has a suite of sensors that give us really fine scale information about what the animals are doing from second to second, actually from millisecond to millisecond almost. And so it's got 3-dimensional accelerometers and gyroscopes and magnetometers. And these are things that are in your phone. So the technology that allows you to shift your screen or shift your phone and the screen shifts with it, those are the accelerometers and gyroscopes that are accounting for gravitational force and other things that realign it. So those same technologies that we use in smartphones are coupled into this. So with that, we can recreate the behavior. We can look at tailbeat frequency. We can see how the animals are modifying their posture in order to go after prey and all sorts of things. So this is really a revolutionary tag that allowed us to go on board with the animal. So the idea is that this will clamp onto the fin. So you can see where it says clamp arm, and then the orange thing is the float, the black part is the camera and logger, and then there's a silver part that looks like a clothespin, and that's basically what it is. So the shark swims by, and and I'll clamp this tag onto the fin as it swims by, and it'll go about its business. And then I will be recording for the next 48 or 72 hours what this animal has been doing. So this is a picture from South Africa. Hopefully you can see it. We'll coax the shark up the side of the boat. Got a little bit shaky for me. I don't know if it did for you all. Let's see if I'll play it again. So coax the shark in, clamp the tag on. Now that shark can swim around for the next period of time, doesn't know it has a tag on it, and do its natural behavior, which we get to study. What's really cool about this is we get to see from the shark's perspective. And so this is one of those key moments that we didn't know occurred until we were able to go along with the shark. And so what we found is that the sharks, so you can see in the bottom of this picture, you can see the shark's head. And we found a brand new, completely new hunting strategy that sharks were using. So historically, we thought that sharks would avoid kelp. They would avoid any of these vertical structures that they'd get restricted in or could run into. And so folks that would abalone dive or scuba dive and things would refuge into kelp for safety from sharks. But what it turns out is that the sharks are actually utilizing those habitats in a very different hunting strategy. So we'll see this shark swim around. You see bubbles come up from the sides. Those are actually seals. There's a seal that pops down on the side there. So the seals are swimming all around. The sharks are actually predating and oh, sorry, there's
the shark swims through the bubbles.
It's a brilliant British voice, but I'll turn it off. So you see the sharks are swimming through the kelp. They're actually pursuing these in South Africa. These are Cape fur seals, actually pursuing the Cape fur seals through the kelp, which is a hunting behavior that we've never seen before. We knew that they did the ambush explosion predations on the surface, but we didn't know that they actually pursued prey in the kelp, which was a new, really new and cool finding for us. And this just breaks it down a little bit to some of the behaviors that I pulled out from that screen. So A is a picture of the shark sees one of the seals as it's swimming, and it'll follow it. The seals start to blow bubbles in some of these other pictures. Then if you look down at picture in F, and the little red arrows actually point to 3 seals, Cape fur seals, that the shark has basically corralled into that kelp patty. And so now it's a different hunting strategy. They're corralling them in there. They're waiting for the Cape fur seals to be distracted. And then they pursue them. So very different than the ambush strategy that we've seen when we had the opportunistic sightings. And in fact, what we find out is if we plotted the movements of that shark, so the black line here is the movement of the shark over 5 minutes. Where it's blue, they're in low kelp, and where it's green, they're in high kelp. And then where you see these red stars, that's places where they actually interacted with the seal. And so you can see from this that the sharks spent a lot of their time, this shark spent a lot of its time in that kelp, somewhere we thought they didn't go, and that's the only place that the shark actually interacted with its potential prey. So again, very different hunting strategy than we had expected or ever seen before. So I know that some of you out there are abalone divers or surfers or swimmers, and this is the type of thing that maybe unsettles you. But I do want to add something else, because what I've also found in this time is that these sharks are incredibly discerning. So what we'll see in this video is you'll see the shark's head in the very bottom, and you can see its tailbeat frequency by how fast its head moves back and forth. So right now it's very slow. It's just cruising at the bottom. It's looking for, it's basically looking for its next prey. It starts to increase its tailbeat. Its head starts swinging back and forth. Now the shark is going full tilt, full acceleration towards the surface, towards an object. So basically that shark has just exerted an acceleration that's 3 times what Usain Bolt can output. So imagine Usain Bolt on the starting line, down in his crouch, about to explode. And at that same moment, you have a white shark that is about 4,000 pounds in water. And the moment that starting gun goes off, that shark has accelerated 3 times faster than Usain Bolt has been able to do. That's what we've learned from these tags, from those data. But what I think is more cool about this is that shark exerted all that energy, got from going a couple miles an hour up to about 20, 25 miles an hour towards the surface, dead sprint, and got to the surface, and it stopped. It got right to the surface where it would attack something, and it bailed. And so we went back and looked at it and said, well, what the heck is going on? It exerted all this energy. It would be like if you were really hungry, ran all the way to the grocery store, and there was this perfect cinnamon bun sitting out in front of you, and instead of eating it, you decided to run back home. That's sort of what the shark did here. So we said, well, why did the shark do that? Because there's obviously something here. But when we took a closer look at this picture, this is a picture from the very peak when the shark, right before it bails at the surface. And it turns out that to my eye, when I saw it the first time, I said, that's a Cape fur seal. You know, that shark's got it perfectly in its sights. It's going to get it. But when we slowed it down and stopped it, it's actually a cormorant. So that the shark, even though it exerted everything, was going 20 miles an hour at the surface, knew that that was not the prey that it was looking for, knew that was not, the cost benefit of going after that was not worth it. So for me, for being a surfer and a water user, it makes me feel much more confident being in the water, knowing that these animals are incredibly discerning, that they oftentimes know exactly what it is that they want and know what they don't want. So after hundreds of hours of video analyzing, looking at different types of prey that these animals could eat, we finally saw a predation. Out of that entire time, one single predation. So these are these mindless killers that people thought were, you know, patrolling our coasts and things. But out of all of that time, from about 23 different animals, only one single feeding event was detected. And I like to show this feeding event because it's the first one I think anybody's natural feeding event from a camera tag anybody's ever gotten. And wait for it. That's it. So that was it. That was the extravagant feeding event. It was a hagfish, which it's basically a slime eel. Has almost no nutritional caloric value. So I think it was just in the wrong place at the wrong time. So incredibly interesting for us to know that after all these hours of tracking all of these sharks, that it's not a feeding fest, that they're actually feeding very, very rarely. We thought, well, maybe there's something going on that the tags are changing their behavior. Maybe they would normally be eating, but the tags are preventing us from being able to see that. So we use a different type of tag to look at that. We use what we call a stomach tag. So this, excuse me, one moment. This is utilizing that cool thermophysiology of the shark. Remember, I talked about that they're able to thermoregulate. So they have a warm, what we call viscera. So their insides, their stomach is warm. So if we put a tag into their stomach, we can look at how elevated their temperature is. But then when they eat, the metabolics of digestion basically increase the temperature. So it's similar to us, like on Thanksgiving dinner. So when you have Thanksgiving dinner and you eat your big dinner, you start to get a little bit of the sweats. You know, you have to unbutton your pants. But that is basically your body metabolizing all that food. And so the same thing happens. The same thing happens when you are a white shark and you feed. Your body temperature, your visceral temperature, stomach temperature is going to increase. And so we know what the shark's average or normal temperature is, and then from the stomach tag we can see when a shark tries to eat something because it ingests a bunch of cold water. So we see the temperature of the stomach drop. And then if it goes back to normal, we know it wasn't successful. But if it goes back to normal and then keeps going up, then we know that shark ingested something with caloric value, and so it's digesting it. So the way that we do this is we take that tag and we roll it up in a little bit of elephant seal or whale blubber. We call it a blubber burrito, and we feed that to the shark. And this is a video that shows just how we do that. We put it behind the decoy. I'll probably have to show it again because it's pretty quick. But we put it behind the decoy. The sharks come up. We pull the decoy out of the way, and it will grab the bait and swim away with that tag now in its belly. Let me see if I can show that again. So again, it just grabs it, swims away. And that will be in the shark's stomach for the next few weeks. We've had them in for up to 2 weeks. And sharks normally evacuate their stomachs. So they eat a lot of like indigestible bits, so bones and other things. So they'll actually just spit out their entire stomach. So at that time, when they do that, the tag comes out, pops to the surface, and then we can go collect it. So these are the data that came from one of those stomach tags. And so this is an example of one of those feeding events. So if you look on the x-axis, so that's time, So 0 is the moment that the tag went in, up to 6 days. And then the temperature, which is in Celsius, is on the left side, on the y-axis. And so you can see it drops down to 22 degrees. That's when the shark ingested the tag. It was sitting in the water. Shark ingested the cold water and the tag. And then we see the little bump up to 26 degrees. And basically, that's showing a little bit of the digestion of the little blubber burrito that we gave them. And then it drops back down to about 25, and it starts to level out there. And then on day about 5.5, we see another big drop in temperature. So that's indication of another feeding event or an attempt. And we know that that was successful because if we follow that on past, we get to about day 6, we can see that again, that temperature is rising above sort of the resting temperature of a white shark. So we know that that was a successful feeding event. Again, so if it was unsuccessful, we'd expect that temperature just to go back up to about 24, 25 degrees. So after over 1,000 hours of data, of stomach tag data from 8 different sharks, similar to the camera tag data, a single predatory event. And this was the significant one, and it was actually this one that I showed a second ago. This was the only predation event that we have a record of from a free-swimming wild animal. We ground truth this by using the sharks that are in captivity in the Monterey Bay Aquarium. So we were able to say that this is really what would happen. But we saw only one event of those in the wild over about 1,000 hours. So so that's a little bit about their behavior and what they're doing along the coast. And so now I want to talk a little bit about what we've learned about their movements and migrations. And so initially, it was thought that white sharks were a strictly coastal species. They were resident year-round along the coast. And to understand that, we want to say, well, is that really what's happening? But we need to be able to, again, go on board with them. Those biologging tags that I talked about before are short-term. Those are only for a few days to maybe a week. And those are more recent events. So we wanted to put on a type of tag that we could track the actual movements of the animals for long periods. And unfortunately for us, for anybody working in the marine realm, a GPS signal doesn't transfer through the air-water interface. Basically, it can't transmit a signal. It takes too much energy. It can't transmit a signal through the surface of the water. So we have to come up with a different technique. So this is an acoustic tag, which sends out an acoustic signal. That signal will send about 700 meters to a kilometer away. And every 60 seconds or 80 seconds, it'll say, I am shark 64 and I am here. And so we set out base stations that will listen for these tags. And so this is just a short video of applying the tag. We put the tag into the dorsal musculature, so right up next to the dorsal fin with a small dart. It goes into the shark, and that shark will continue to swim around. And now it has a tag that will stay with it for the next 2, 3, up to 5 years. And every 60 to 90 seconds, we'll emit that acoustic ping that tells the receiver that it's around. So what we get from those is some resident data. And so this is information. So along the x-axis, again, time, and then the y-axis there is the detections per day. And basically what you take away from this is that over a period of 4 years, we can see that from October to March every year, there's these peaks in the numbers of detections. So that's the residency period of white sharks along the coast. They are peaking in sort of that late fall, early winter period. The colors are just the cohorts of the years that animals were tagged, so that's not a big deal. But just notice the general shape. So it shows us those animals are predictably arriving at our coastal locations during those months of the year. But the big question is, well, that's great about when they're here, but what happens during those spots from April through August where we don't see any data? Where are those animals? And so the thing about acoustic tags is that unless you have it near a base station, you can't tell where the animal is. So it doesn't give us any information about the shark when it's not around our receivers. So now what we need to do is come up with a different technology that could actually track sharks when they weren't where we were predicting them to be. So this is what we call a satellite tag. And the satellite tag is a little bit different. So instead of transmitting the information to a base station about where the animal is, it is actually going to archive the information. So it's basically like an onboard computer that records temperature, depth, and light level. And as I said before, GPS can't go through the air-water interface, so it doesn't have a GPS. But what's really tricky about it is that it uses the light level. So you're probably familiar with the idea of Greenwich Mean Time. And that was really established so that when you were a seafarer, that you could reference your local noon, the local time of noon where you were, to Greenwich, and you know where you were basically on the globe. So where I am here in Oregon, I know that if you take the midpoint between sunrise and sunset, that is my local noon. And if I compare the offset to that, to GMT, which right now is, I think, 7 hours, I would be able to figure out where I was on the globe. And so using that, some temperature and depth data, and doing some fancy other math, we can basically estimate where the animals are. But the caveat about this is that it's within, because you're using light levels and there's some sort of fudge factor in there, that it's not accurate to the degree that we need it to be to do fine scale movements, but it gives us an idea of their oceanographic movements on a large scale. So it has about a 200 kilometer error on it, but when we're talking about global movements, that's doable. So this is I'll show you a picture or a video of what we found when we put the satellite tags on. And as you can see, these animals are moving from the California coast offshore to the spot we call the White Shark Cafe, which I'll talk about in a few minutes. Some are going to Hawaii, and then they're all coming right back to the coast. So this is what they're doing during that period when the sharks are not available to us at the coast. They're moving offshore to the spot in the middle of the Pacific, and then they're coming back every fall. So our big question was, well, what are these animals doing? Why are they leaving the California current, which is basically it's an all-you-can-eat buffet. So imagine that you're in Las Vegas and you're at the buffet and you're just eating crab legs, you're eating steak, you're eating everything that you could possibly want. That's basically what the California current is. And these sharks are going from there out to the White Shark Cafe, which for all intents and purposes, from what we know, it's basically an oceanographic dead zone. It's like going out into the middle of the desert in Vegas. So you're an animal that's at this wonderful feeding buffet, you're going out to the middle of the desert. Why would you be doing that? And so there's only two basic drivers that we could think of why animals would be going there. One is for food, so it's an elephant seal on the top left there. There's something else out there that they are trying to eat. The other option is that they're going out to mate. So we wanted to start to look into this a little bit more. So how can we understand what the animals are doing while they're out there? So the old adage about you are what you eat really holds up, and we can actually study that. And so if we look at this figure, this is obviously the Northeast Pacific Ocean. The color is the productivity. So the brighter red is more productive, and the darker the blue is the less productive. So you can see, imagine a shark is on the California coast and it's feeding on things in the really productive coastal waters. And so we'll call those prey, the signature of those prey green, right? So if you move off into the offshore environments, the prey that are out there, those are blue. The isotopic signature of those are blue, because basically the primary production in different locations has different ratios of isotopes. And isotopes are just basically, they are different versions of a molecule, basically. So we look at carbon, or an element. So we look at carbon and nitrogen. And so if you are a primary producer on the coast, your carbon and nitrogen isotope ratios are going to look different than a primary producer out in the offshore. And then that goes up the food web. So whatever eats them is going to look like their signature, and so on and so forth. So wherever you eat, you're going to look more like those prey. So the green shark looks like the prey on the coast. The blue shark looks like the shark in the offshore environment. And if you were to eat at both places at the cafe, it would be a mix of those two things. So we went and studied took samples of all the different potential prey species of white sharks at these different locations. So we have pelagic species, which is offshore. That would be the cafe. We have Hawaii and then California. And so we looked at their isotopic signature. So what were they made up of? And then we said, if you were a white shark and you were made up of equal parts of all of those, if you were going to feed on all of those equally, then you would be right in the middle. But if there's a better resource in California for feeding, you're going to be closer to California. You're going to look green. If you are going to the cafe just to feed, then you're going to look more blue. And so when we analyzed the actual data from white sharks, we found out that they're actually closer to California. They're in the greener side of the blue-green scale. So it showed us that the sharks are not going out to the cafe to eat. That's not their main purpose for going out there. And this was followed up by a study we did about buoyancy with them. And the thing about sharks is that they don't have a swim bladder like other fish do. So they can't regulate their buoyancy by inflating or deflating that swim bladder. But what they do is when they eat, they absorb all of the lipids into their liver, and then that oil is lighter than water, so that helps them to be more buoyant or less negative. And so if you're a really well-fed shark, then you have a big liver and you are a little bit more buoyant or less negative. So when you're at the coast, you're filling up on food, you have this really robust liver, so you should be a little bit more buoyant. And we know as they're moving out to the cafe, they're doing the same thing that a bird does when it flies long distance. So the shark is going to swim towards the surface, and then it's going to glide down the same way a bird does it, same way it swims the surface and glide down. It's a very well-used technique to conserve energy throughout the animal kingdom. So birds do it, elephant seals do it, sharks do it, and it's a way to get from point A to point B with less energy. So if you're a shark and you're swimming up and you're gliding down and you're really robust and you have a lot of lipids, then your angle is going to be really shallow, right? So you're going to start, because you're going to be pretty well buoyant, you're going to be almost neutral, you're going to fall very slowly. And as time goes, if you burn those lipids, if you burn all the energy that's in that liver, that you're eventually going to get more and more negative. So you're going to start falling steeper and steeper and steeper through time. And so these are two predictions. One is on A is you're really buoyant. That's sort of your glide and your sort of your glide angle. So on the left side is really it's shallow, and on the right side is you get less and less energy, or you use up more and more of your energy. You are going to basically get a steeper and steeper glide. And that's what we found. And this is just your drift rate. So that's how fast you're going down over days into migration. And what we see is as you go through time, once the sharks leave the coast and they start going towards the cafe, you can see that they are, their drift rate is increasing. So that's indicating to us that they're losing body condition. Basically, they're not feeding. There isn't a better resource out there that they are exploiting. We can also see this when we see the sharks on the coast. This is a shark, well-known shark called Mr. Burns. And he is, this is a picture of him about 2 months apart. On the left side is when he first arrived. And on the right side is a few weeks later after he ate a few elephant seals. And we can assess his body condition by comparing the height of his dorsal fin to the girth of his body. And by comparing those ratios, we can look at how he's basically, we call his body condition has changed over time. And so if we look at this figure on the bottom, again, it's over a period of time. And this is a number of different sharks that we got pictures of week 1 and week 3. And we can see over time that all of their relative girth rates went up. So they all were putting on girth during the time from the moment that they got to the coast to the moment 3 weeks later after they had fed some. So that was an indication to us. Another indication supporting that these sharks are not necessarily eating offshore, but they are likely doing something else. I am going to plug my computer in before I lose you all. I thought it would make it. So I'm going to let you think about what they might be doing in the cafe for 2 seconds while I plug in. And this also gives you a chance to have a quick sip of your drink if you hadn't been doing it already. Okay, we're back. All right. So what are those animals doing when they're offshore? And so this is a figure of their vertical velocity. And the part that I want you to look at is that sort of April to August period, the part where the vertical velocity is very high. So that is the time when the sharks are in the cafe. And vertical velocity is basically how fast these animals are going up and down on the water column. And so you can imagine that as your shark is drifting down and drifting up, it's moving up and down on the water column. Well, that's November through March. It's very minimal amount of movement up and down on the water column, very slow. But when you get to the middle of the cafe from that April to July region, that increases incredibly. So these sharks are actually powering up to the surface and down to 300 meters about 150 times a day. So that's basically, like running sprints, 300-meter sprints 150 times a day. And so that's happening exactly in the cafe. And so when the sharks are centered there, that's when the rapid oscillatory diving happens. And as they move further away from the center of that cafe, it starts to tail off. And so for us, this is an indication that this was something to do with perhaps something to do with mating. And the way that we think about it is that some shark species are known to release pheromones to present sexual readiness. And so when you're out in the ocean, it's not a uniform medium. Basically, the ocean is like pancake layers. So you have temperature, you have salinity, or something else that is driving these sort of like layers in the ocean. And so if you're a female and you release the pheromone at this layer, it wouldn't distribute throughout the water column. It's going to stay in that layer. So if you're a male that's able to swim up and down through those layers, cut as many pieces of those pancakes, you're more likely to find that female. I guess you have to think about it in terms of a stack of pancakes. You only put syrup on one of the pancakes, and you have to be able to, you want to take a bite and make sure that you get syrup. The only way to do that is to dig all the way through it. Anyway, so we think that maybe the idea that the fitter male, the stronger male, the one that's able to feed better on the coast and get out there, has a higher likelihood of interacting with the female because that male can do more dives and more for longer periods of time. So a quick summation of that is that the sharks are along the coast, hopefully feeding or likely feeding on marine mammals. They're going off to the cafe to likely mate. So that's a little bit about the movements and the migrations. I've got about 10 minutes left, and I just want to talk about one kind of cool aspect that we've learned about some of their interactions with other species. And this is a distribution of their, or a picture of their distribution, as well as their prey and what we call an intraguild competitor. And so the prey is the blue, that's the elephant seal, the green is the white shark, and then the gray is the killer whale or orca. And so killer whales and orcas, they're what we call intraguild competitors because they fill the same ecological role as a white shark. They're eating some of the same prey, they're about the same body size, and they're in the same environment. So they have a potential to overlap with white sharks. So on the left side there is basically a frequency distribution at the coast. So you see from March, which is month on the x-axis, so March to December, you can see the top one, the blue, that's the distributions of elephant seals at the coast. You can see that the number of elephant seals peak in April and March, and then they peak again in the fall. White sharks seem to peak only in the fall, and then the killer whales, the orcas, peak again in April, May, and then in November, in October, November. So there's a spatial overlap of all of these different predator, prey, and competitor at the same time in the fall. And what we know now is that there's three different, on the West Coast here, there's three different ecotypes of orcas. There's the resident killer whales, which are the type that are eating mostly salmon, the types that you find in the Puget Sound. There's transients that are focused more on the marine mammals. These are the ones that are eating seals and sea lions and whales. And then we have the offshores, which are a group that actually focus on deep water sharks. And so it's pretty cool because you can look at the dentition. So in C, where it says offshore, those are the teeth, and you can see that they're ground down. So those sharks those orcas are feeding on sharks, and sharks have this really coarse skin. So the orcas, as they handle them, basically are sanding their teeth down. But they're killing sharks, and they're eating their livers, basically that really rich, lipid-rich part of the shark. And so at the Farallon Islands, where we work, all these animals interact at different times, or interact almost simultaneously on occasion. And so on the right side of this, and what you see is figure D, you see that's again that map. And what I want to look at is the orange, which is the Farallons, and then the green, which is Tomales Point, and the blue, which is Año Nuevo. And those are the receivers that we have in the water there. And on the left side, at A, if you follow that through time, those are the detections per day. So that's basically equivalent to like the numbers of sharks that we're seeing at those locations, and those colors coordinate with the locations. So you can see that normally in October and November, the number of sharks at the Farallons basically peak. and then they drop off, and then the shark numbers peak at Año Nuevo, and they sort of hold steady at smallest point. So that's the green. But what happened in 2009 is that killer whales came through, and they actually transients and offshores, and they killed a bunch of marine mammals. And at that point, there was this huge exodus of sharks. So this is, B is now 2009. And I just want to compare that to the previous one, which is an average. And so you can see in November, the day that it happened, within, I think it was 5 hours or 7 hours of the moment that the orcas came through. They were there for less than a half an hour. The moment that they came through, all of the sharks in the Farallones left. And they distributed themselves to other sites along the coast. And then only one shark came back for about 5 minutes and left again. So the whole rest of that season, when sharks normally stay around for about 37 days on average, the sharks curtailed and left at that moment that the orcas came through, and none of them came back. One of them came back for a few minutes for the rest of the season. So it's really incredible. So basically those orcas, even though we don't know that they killed a white shark, and they definitely didn't stick around for more than a few minutes, but that was enough to displace those sharks completely from that area for the whole rest of the season. The next year, everything restarted and was back to normal. That very similar to what you see in the A figure on the top. And so this is really, I guess, really important for white sharks, but it's also really important for their prey, for the elephant seals. So again, this is a peak time when elephant seals there. And this figure is going to showcase it, but I want to set it up a little bit. So on the bottom, you see the mean fall seal count, and then the predation rates of sharks on seals. And so basically, when you're at the Farallones, the number of seals every year are a little bit different, but the number of attacks or predations on seals usually is pretty tied to that number of elephant seals that are there. So it's almost a linear or straight relationship. So that's going to be represented here. So it's a little bit busy, but I want you to focus on the black dotted line in the middle. And so that is the actual rate, the relationship between the seal count and the predation rate. So you can see it's a straight line, and it's increasing as seal numbers go up, so do the predation rate. But then I have these other dates that are listed here with the triangles. And those are years that orcas were seen at the Farallon Islands. And so basically what happened is that the orcas came through, displaced the sharks, and that displaced that predatory presence or effect of white sharks on elephant seals. So elephant seals basically got a big release of pressure from the white sharks because the orcas displaced them. So it has this, what we call a trophic interaction. So it's on these different levels of predators and preys, that this effect was working over. And so you see in 2011, they're sort of up closer to where that black line is. And that's because the orcas came through later on in the season after basically all of the sharks had mostly left anyway. But it's really cool to see that there's this big effect from orcas coming through, whether they actually ate and killed animals or if they just displaced them by being present. So that's a little bit about the work I've been doing. I'm continuing that work here in Oregon. These are just some figures of some tracks of sharks on the left side that have gone out to Hawaii in the cafe and come all the way up here into Oregon. And on the right side is all the gray circles are places where sharks have been detected. Not the white circles, but the gray circles, and the numbers of those that have been detected up the coast. So my goal is to study how the animals are moving here and how climate change is basically affecting them. And this is just a few months ago. This is a juvenile sperm whale that was here on the coast. And it was a good indication to me that the sharks are here and are here in fairly decent numbers. I counted about 48, I think, different bites on this animal. Sorry, it's a little gruesome for anybody that's eating right now, but just a good indication that the sharks are really still present here in a pretty big way, which is cool. So hopefully we can learn more. We know almost nothing about the sharks when they're here. So with that, just a quick summary, that shark attacks are rising, but the likelihood of being attacked is decreasing, so your individual likelihood is going down. Their abundances globally and off our coast here are pretty low, and they're seasonally here coastal, but spend most of a good period of their time offshore. And we think that they're here to feed and they're offshore to mate, but we're still working on that. And then I'm hoping to look at some more of the movements of the sharks here and how they're the consequences of climate change and warming waters, which we're already starting to see. And with that, if you want to learn more about some of the research I do, there's a couple shows that I did with Discovery, so the Great White Highway and Blue Serengeti, a couple more with National Geographic. But with that, yeah, I'm happy to take questions, answer anything I can. My email address is up at the top here, my website for my lab, and then my Instagram if you want to keep up with some of the, when we're actually back in the field doing work with some of the work and things that we do. And if you have questions and things to ask me later on, I'm happy to answer any of those. So with that, Cinnamon, what did I miss in the chat?
Sure. Go ahead and type any questions you might have into the chat, but I'm going to start back where we were at when we first started seeing questions. So the first one I have here is, what is the average length of a white shark, and how does that compare to a 70-year-old shark?
That's a great question. So we don't, an average length, well, the average length actually depends on where you are sort of around the globe. So I would say the average length off the West Coast here is probably in the 12 to 14 foot range. You know, they start out at about 4 feet. There's probably a bottleneck sometimes somewhere around the like 7 to 12 foot range where they're probably competing with adults, with big successful adults. So there's probably not as many of them make it through that size, but we probably have most in that 12 to 15 range, and they get up to somewhere about 20 feet as the largest actually measured animals.
Nice. There's another question here about some of the work that you were talking about the sharks foraging in kelp was in South Africa. They're curious on whether you're seeing that same kind of behavior in California and whether any of the abalone divers have witnessed that behavior.
So I don't know that anyone's, I know that the abalone divers have seen them around kelp, but I don't know that necessarily seen them in the kelp. But that might not be a detail that any of us have really tried to tease out. With California, what's been interesting is that recently we've had these big die-offs of kelp. And so the camera tags have only been around since 2013. We started putting them out in California in about 2015. And so by that point, kelp forests were already pretty well diminished in regions. And so we haven't seen that much. Although this year, I should say at Año Nuevo, one of the spots that I spend most of my time, we had an incredible kelp year, kelp places I've never seen, which was unlike anywhere else along the coast. And we actually saw the sharks were not in their normal spots, but they were hanging out right on the edge of a kelp bed, basically where the elephant seals would come off the shore and then channel through the kelp. So whether they're going in and out of it, I don't know, but they were definitely utilizing it at that point in California.
Interesting. We also had a question on whether you had a camera that was shown and we were able to see the shark's head moving. The question was, is there a camera or an ability to remove that shaking so you get a better image? Not
really. That's sort of one of the factors. You could place it a little bit further back, but you're still going to get some movement in the shark, because basically when they really start to move, they've got one pivot point, but everything is moving. Their whole body is basically undulating. It's the type of movement that they have, and so it's almost impossible to really find that critical point. And for us, it's really important to not be at that critical point, because we want to look at their tailbeat frequency and the amplitudes of that, so we can look at the amount of force that these animals are actually exerting in order to do their behaviors.
We also have two questions about the distribution data. The first one is just, where is that information, that spatial data available? Is it available to folks that are doing some work with offshore energy planning and those kind of things?
So the beginning of that was the distribution data of the sharks and where they're. Mm-hmm. Yeah. So most of the data, if you go to topp.org, which is top.org, it's the tagging of Pacific pelagics. A lot of the data is showcased there. It's also the part of the Census of Marine Life, the whole TOP program. All those data sets are publicly available. So you can go through and actually get the raw data for those if you're interested. If you want something that's more filtered and actually presented, then that some of that is available on the top website. If there's something else that you're looking for, I'd be happy to sit down with you and go over some of the more specific things, because there's, we've got about 500 tags that we've put out, so you might not want to leaf through all of those.
So that question came from Rebecca. So Rebecca, if you're still online, if you still have questions, go ahead and contact Taylor directly. The other one related to the tracking data was, is this information used on oceantracks.org? It
is, absolutely. So that is, if you poke around, you actually see me giving a couple brief webinars in Ocean Tracks. So yeah, so this is the part of the top program, which we looked at whales, birds, turtles, sharks, all sorts of different species. And so those are the data that are showcased on Ocean Tracks. And so for anybody that has kids or curriculum and stuff, Ocean Tracks, so oceantracks.org is an awesome website. It's got curriculum built in. It basically uses these data, albatross, bluefin tuna, and elephant seal data, our real data, and has a bunch of different techniques and toolboxes that you can use to analyze data. It's for students from high school up to college. So it's a really cool resource if you have students or kids at home that want to sort of geek out a little bit about science and the oceans a little bit more. So thanks, Rebecca, for cueing me on that one.
So another question here about your tag data. It shows one predation event every 40 days. Is that really what we think is happening with them?
Yeah, and so back in the 80s, there's this incredible scientist named Frank Carey that did a study, just crazy study, that he was able to estimate the metabolic rate of a free swimming shark with a thermistor in its body. And he estimated that a 30 kilogram chunk of whale could last a shark up to about 6 weeks. So based on just like what its routine metabolic, what it needed to stay alive, basically. So we expect it to be really low. So it has to do with like they're incredibly efficient swimmers, and they're also maybe really efficient at digesting. And then again, that they're taking bulk on. So it might be that, the sharks are eating more than like 30 kilograms over 6 weeks, but that's just what we think they probably need to maintain. So this fits well. Actually, it's crazy though. If you take all the numbers of hours of video and then numbers of hours of stomach tag data, put those all together, it actually comes out to be almost exactly 6 weeks. So it sort of, the math works out in an odd way that we have that one real predation in 6 weeks of different types of data. It's not on a single animal, but it's sort of cool how that lined up.
We also have a question about how do you find white sharks off the Oregon coast, and is there a way to be involved in shark research here locally?
Absolutely. So yes, to the second half part of that question, feel free to email me. I am just getting projects started, so would love to have local help and local contacts and folks that are willing to come out. And finding sharks is going to be a little bit tricky. So basically what I'm doing now is I have a lot of data from the sharks that I've been studying in California for the past 15 years. And so I'm modeling the types of habitats that they use there. So the temperatures, the dates, the marine mammals, the coastlines, all those things, figuring out what they're choosing there, and then trying to overlay that over Oregon to figure out what places that would line up, and lining that up with sightings and attacks and all these different characters. And I'm also trying to get tags and things in sharks, more tags along the coast, or more sharks tagged in California, and then more receivers along the coast here to see how the sharks should be using it. Because the locations that I've seen the sharks detected along the Oregon coast are other folks that have given me data that they've had. So I'm hoping to know more, in the next year about where these animals are.
Do you know the age structure of the population of the 219 sharks?
We don't. And that's, some folks tried to extrapolate an age structure. And so for those of you that know, an age structure is basically the age breakdown. So how many animals size, of age 10, of age 11. One of the big problems with age structure is that we don't have a good grasp on the age to length relationship. So if we see, I think what happens is as you get older in a shark, you sort of, what we call asymptote. So you sort of get to that maximum length that you're going to get, but we don't know how fast each individual reaches that. And so I have a shark that grew 7 feet in 4 years. And I have other sharks that have grown 2 feet over the last, about 30 years. So we don't have a real good estimate, and they were at the same size. And those sharks started at the same size. So we don't have a real good estimate of the size to age ratio or relationship. So drawing an age structure. We have size structure, but we don't have age.
And how do you age those sharks?
So the aging sharks, historically it was through the vertebrae. They lay down rings, basically the same way that a tree would lay down rings. But that gets really confusing as you get older. These long-lived species, the rings get really close and things get confusing. So now they use bond carbon dating, which is basically they look at the carbon isotopes that are in the vertebrae of the shark, and they can extract those out, and they can look at how the carbon signature was laid down in the animal over its lifetime. And then they can look at the carbon isotopes in the atmosphere that were a result of nuclear testing in the 50s. And we know what that looks like, the carbon concentration in the atmosphere. And then we take this shark, and its carbon signature looks like this, and then we figure out where that fits with what we know about the atmosphere, and we can say, that this shark was born in 1963, because it matches 1963 until now. So that's how we've been able to age them up to 70 years.
Nice. Can you tell us a little bit more about how you decided to focus your research on sharks?
Sure. I got originally interested because I started working on predator-prey dynamics. And so I started working in Lake Victoria with Nile perch and cichlids. There's really cool predator-prey dynamics that was changing the habitat of the entire the entire lake ecosystem. And then sharks were sort of the next step of a group of animals that were really important in terms of their predator-prey dynamics, and very little at that time was known, and even now still not that much is known about them. So that's how I got started in sharks, and I've just been fascinated by them. I've been scuba diving since I was 13 and have loved to be in the water ever since. And sharks are one of those things that just, I think you're all here listening as well. They're cool. They're exciting. Nobody's here to see me. You're here to hear about sharks. And I also wanted to do a job that was cool and challenging every day. And I didn't want to go to work and sort of regret getting up in the morning. So every day I go to work, I'm really excited because either I get to work with sharks or I get to learn about them. So that's why I ended up with them.
Nice. Do you anticipate any difference in shark behavior around Oregon compared to the places you've studied in the past?
Do I anticipate any different behavior? That's a really interesting question because yes and no. I think that right now, I think it's going to be a little bit different because I think that the water is cold. Well, the water is colder here, obviously, than Central California. It's 2 or 3 degrees Celsius colder. And so the sharks aren't going to be able to hang out and aggregate in the same way that they can in California. Basically, they don't have it takes too much energy. It's like hanging out outside when it's 30 degrees as opposed to when it's 70 degrees. Like, you just can't hang out outside that long. You can do it, but not that long. So I don't think we're going to see the same aggregations initially. But I think as climate change continues to change water temperatures, and as we start to see these warm water blobs moving up, as we have since 2015, I think we may see more sharks starting to establish. So initially, I think we might see sharks that are moving more than being aggregated. The Farallones are there for 37 days on average. So I think we won't see that yet, but as the water temperatures warms and the sharks can stick around for longer, they probably will.
Can you tell us a little bit more about how sharks communicate with each other?
I wish that I could. So we know that there's a couple different ways that they do body posture. They can signal different types of situations from body posture, pheromones. I'm guessing this is going back to perhaps the question about the orcas, the killer whales coming through. And that is a big question for us right now, is that we're trying to figure out how. So the Farallones is about it's about 1 kilometer across. And so there's sharks on one side of the island, sharks on the other side of the island, and currents aren't necessarily taking the water there. And so how animals on the far side of the island knew within a few hours that there were orcas on the other side of the island, and that there was some predation or whatever was happening. And the cool thing about transient orcas is they're the ones that go silent when they hunt. So there's not a sound that they're hearing. hearing. So the orcas go silent when they hunt the marine mammals, so they can't hear them. So the sharks couldn't hear that either. So there's some type of communication that's going on that we don't quite understand yet. So there's a couple different ideas, but you know, we haven't quite figured out how they really communicate on that broad of a scale.
And then I think this will be our last question, but as a shark researcher, can you still enjoy watching films like Jaws and Mega, or do you just spend all the time picking them apart?
Oh, I should. I would get up over here and grab the booklet of DVDs that I have. So my wife Jamie and I, she's an ecophysiologist, also at OSU, and we met in grad school. And from that moment, so grad school is when I started working with white sharks. So from that moment on, we decided that we were going to watch every bad shark movie that's ever made. I guess I should say every shark movie that's ever made, because 99% of them are bad. So yes, I love bad shark movies. It took me a while to get through the Sharknado one, but I think they're really entertaining, and there's way more than you could ever imagine are out there. But yeah, no, I can still watch them, and you know, I can watch them for the value that they had at the time. And I saw Peter Benchley talk during the Summer of the Shark, which was like 2004, maybe 2001, something like that in Massachusetts. And he really regretted what Jaws did to an entire generation. And you know, I agree with that it really changed people's perception. And I guess the things that I can't watch are when like the Meg, like that's hilarious, right? It's ridiculous. Sharknado, it's ridiculous. Sharktopus, all of those. But when folks started to dive into the realm of trying to be scientific but also sensational. So you know, some more of the popular media documentaries that follow science, that really portray themselves as scientific but really sensationalize what sharks are. And not really telling the true story, but trying to sell a product. I have a really hard time watching those. And in fact, I won't watch those. But the ones that are a bit ridiculous and you know that they're ridiculous, I'm a big fan of those. If you need any recommendations of bad shark movies, let me know.
There's a few other more detailed questions. I'm going to let those folks go ahead and reach out to you specifically to get the information that they need. For those of you that are with us, I just want to thank you so much for being a part of our virtual Science on Tap. This is not the way we normally do things, so I thank you for joining us, and hopefully you enjoyed this talk, and I hope that we get a chance to see you in air quotes, at our next virtual Science on Tap on June 25th. And Taylor, you can't see it, but I'm sure all of us are giving you a huge round of applause and thanking so much for being a part of this and spending your time with us. So thank you.
Thanks. Thanks everybody for coming, and thank you, Cinnamon, for putting this on. And again, if anybody has questions, feel free to reach out to me. I also on my website have a shark ID page and shark sighting. If anybody's in Oregon or on the West Coast and wants to add that information of any shark you see, please put it up there. That'll help us with our research. And yeah, I hope to someday sit down and have a real beer with all of you. So all of you that are of age, that is. So wonderful to have you all here. So thanks so much.
Thank you so much. You're getting lots of thank yous in the chat box. And to everybody else, that's a great way to be involved in research, is if you see them, make sure we know about it. So again, thank you so much. And thank you everyone online.
Thanks, Cinnamon.
Bye now.